Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the lecture number 21 and today we'll discuss about high content imaging. So what is high content imaging? So you already know about the 3D imaging and 4D imaging of any sample that we already discussed in some of the previous classes. Okay, so you can do multicolor imaging for any sample, and also you can do multi-dimensional imaging like X, Y, and Z imaging you can do for your cell or the tissue sample. So high content imaging is nothing but you will image thousands of samples together. Okay, so it's the same method, the way you image a single sample in 3 dimension or 4 dimension in multicolor, multi-channel. Okay, so the same thing you have to do for thousands of samples. So when you need to image only 1, 2, or 3, or up to 10 samples, you can go for the manual sampling. You can change the slide, or you can change the imaging vessel on your own, one after the another, and you can go for the imaging one after the another, right? But when you have to do the imaging of thousands of cells or thousands of different samples, let's say treated with 1,000 different compounds, then you cannot do it manually. And even if you go for manual experiments, it takes a lot of time. So for that, we need the automation, in which if you prepare your samples in, let's say, 96 well plate, the way it is shown here. Okay, so each well has the same cell, but different compounds are added to this different well. So you can see the effect of so many different compounds in the same experiments. Okay, and then the microscope is automatized in such a way so that it will move from one sample to the next sample, second sample to the third sample automatically, and it will capture several images from these individual wells. Okay, and then it will have the sophisticated features for image analysis and data analysis. So it can give you the direct statistics that, okay, out of so many samples, this many samples show some kind of effect on the cells for this drug. Okay, so high content imaging is an automated microscopic imaging and data analysis method for analyzing thousands of samples for screening the effect of thousands of compounds, maybe small molecules, peptides, or RNA interference on cellular phenotype. It's a phenotype screen basically. Okay, so you treat your cells with different compounds, and then you capture the images to see the differences in the phenotype. Let's say phenotype means whether the cell shows the higher expression of certain genes or lower expression of certain genes in the presence of these compounds, or how the cell division is affected, whether in the presence of this treatment it may be possible that compound may inhibit the cell growth, so cells may not divide in the presence of that compound. Okay, so those kind of phenotypic information you can gain from this high content imaging experiments. So there are multiple names which are generally used for this technique. Sometimes it is referred to as high content imaging, high content screening, automated imagers, high throughput microscopy, or image based screening, or sometimes cellomics. Okay, you must be familiar with the proteomics, transcriptomics, right? So similarly, this approach is known as cellomics, and its major applications are in drug discovery, screening libraries, large tissue imaging. Okay, so don't try to use this technique when you have hardly 10 to 15 or even 20 or 30 samples in hand. Okay, for those kind of samples you should always go for the manual imaging, the way we go for 3D or 4D imaging for samples one after the other. But when you have more than 100 samples, let's say, or more than 1,000 samples, then you need to go for high content imaging microscope. And generally it is used for fixed cell analysis, for immunocytochemistry, immunohistochemistry, or fish kind of experiments. You can go for high content imaging. Generally it is not a suitable method for live cell imaging, because the microscope takes some time to image the sample which is present in the first well to the image the sample which is present in the 96th well. So it takes few hours to image all the samples, right? Now if you go for the live cell experiments, uh, uh, the time required for imaging one plate will place the cells to other stage of the cell cycle for the next place and for the next plate, right? Because it will take at least few hours to image one plate. So live imaging or kinetic measurements can also be performed depending on the time scale. So only if your live cell imaging experiments, you are not imaging some fast dynamics or fast biological processes, then only you can go for the live cell imaging using this technique. But generally for the fast dynamic things, it is not recommended because it takes some time to image one plate at a time. And this is how the high content imaging microscope looks like. It looks slightly different than your typical fluorescent microscope. It looks like a box, but everything like the objective lens, the other microscope optics, and the filters and lasers and everything is everything is gathered in this box. And this box also has the robotics, so that you have to load multiple 96 well plates in the box as a sample, and then it has the robotic arms which will take this first plate, and the microscope will image all the samples one after the another. Then it will automatically move this plate from the stage, and it will take the another plate and put it on the stage for imaging another 96 samples. Okay, so automatic robotic arms are required for changing the plates on the stage of the microscope. So how this thing works? So suppose you want to grow, you want to do some phenotypic experiments on certain cell line. So you grow the cell line in this kind of 96 well plate like this, and the number of wells you have to decide based on your experiment. Not necessarily that every time you may have to go for 96 well plate. You may do it with 48 well plates as well, or even you can go for 392 well plates. Okay, so depending on your throughput, you have to decide that what plate, which type of plate you will use. So here, for example, we have shown the 96 well plates. So you grow your cells in this multiple 96 well plate, then you add your compounds to all these wells in different plates. Okay, and generally this experiment is performed in triplicates. So you have to add the same compound in three different wells or in three different plates at the same position, so that during data analysis, the software will automatically count the phenotype for those three wells, and then it will give you the average or the mean and the standard deviation for those samples. Okay, so you have to define in the software that, okay, these are my three triplicates for this sample. Okay, and then after adding the compound to so many wells, you just incubate your plates for whatever treatment time you want to give, maybe 1 hour, 2 hour, or maybe 24 hours. Okay, and then you put all these plates in the microscope. So automatically the robotic arms will take the first plate, it will image all 96 samples, then it will take the second plate, it will take image another 96 samples, and then it will take third plate, it will take image another 96 samples. Now how many images it will capture per well? Okay, so here you can see image frames in a well. So in one well, this microscope will take the images like this, 5 images or the 5 filled this way, and 1 2 3 4 5 6 and 7 filled this way. So from one well it will capture 35 fields. Okay, so you can see this is just one field. So this is the layout of one well. In one well there are so many fields the microscope has captured. Okay, and here they have enlarged one particular field. So here you can see the DAPI stained nuclei, then phalloidin stained um, uh, another protein here, and the paxillin stained protein here. Okay, so different dyes or different colors you can use for labeling different proteins in your sample, and also you can stain the nuclei using DAPI. Okay, so this kind of images, 3 color or 4 color, and in Z-stack even, it can capture for multiple wells and multi positions per well. And then the software offers the feature for automated image segmentation. So software can segment the image and count the data automatically. So as you can see in this image, the software will automatically detect the nuclei and the cell boundaries. Then it will detect the actin fibers based on this phalloidin staining, and then it will detect the focal contacts based on this paxillin staining. Okay, and then it will give you the statistics per cell, that number of cells in per field, then the cell morphology, actin intensity or fiber length or the numbers, and the focal contact, focalization, intensity, and the numbers. So this way, all this data analysis is mostly automatized in the software. So it will give you all these numbers. Now here I have shown one image of the software that is generally used for high content imaging. So the name of the software is Cell Pathfinder for high content imaging, data analysis, and data visualization. Okay, so this is just to give you one example there, but there are several other commercial softwares are also available for the same kind of analysis. But you can see how this software look like. So here you have the option to select the specific plate, specific well and all, and then so you can select what are the well you want to see. So for this well, it must have captured several images, but here you are seeing just one out of it. Okay, and software automatically detects the nuclei. It automatically segment the cell, and then it gives all the statistics about if you want to measure the fluorescence intensity in each of these nuclei, if you want to get the idea of the area of each of these cell types. Okay, so all statistics, whatever you want to measure, that the software can measure automatically. And then you can see it gives you different types of data visualization options also. So you can represent your data using heat map or the pie charts or the scatter plot. So all these options you have, you just have to select that how you want to present your data. So here you can see many samples, easy replicates, and equal treatment. So if you go for the 96 well plate, so you can go for the 4 into 8 point dose curves in triplicate. If you have 64 into 8 point dose curve, then you should go for the 1536 well plate experiment. Okay, so automated microscopes often have a long working distance objective to provide versatility. Okay, so generally this kind of microscopes, which are generally used for high content imaging, they generally avoid using oil immersion or water immersion objective lens, because the objective has to move automatically from one well to other well. So still that is technically little difficult to put the oil every time or to put the water every time between the objective and your sample chamber. Okay, so that's why generally the long working distance objectives are used. So the objectives can image the sample from a distance, and it can move from one well to the other well automatically without any human interference. Now, for this kind of experiments, adequate cell density is very important. So when you grow your cell, you have to make sure that they are just grown to the perfect cell density. They should not be overgrown or they should not be undergrown. So if you see, if your cells are like this, the confluence is very high, it is a dense culture, and then the segmentation will be very difficult for the software. Okay, so ideally this kind of cell density should be there. So this, which is known as the sparse density. So around 15,625 cells per centimeter square. So this is an ideal cell density. So software can easily segment this kind of cells and give you reliable data. Also, it has been shown that uh, in this kind of cells, very highly dense cell population, the reactive oxygen species also changes, and also the mitochondrial membrane potential changes when the cell density is very high. So those kind of intrinsic parameters may affect your results. So that's why you need to adjust your cell density very critically. Okay. Some of the practical considerations for high content imaging experiment setup. So regarding plating and preparation, so how many conditions do I need? So and include controls. Okay, so on top of your conditions, so let's say you want to screen 1,000 compounds, so you will have 1,000 different vials in triplicates and plus some controls. Okay, also you have to define the appropriate seeding density for your assay. For specific assay, the seeding density may be different, so you have to optimize those things. Then what is the time scale? So most of the time this technique is good for imaging fixed cells. So if you are imaging fixed cells, then the time scale really doesn't matter. Okay, but if you are using it for live cell applications, then time scale matters, because let's say if the microscope takes almost like 5 hours to image one plate, and then it will go to the next plate, so there will be a time difference of 5 hours between your plates. So in 5 hours maybe the cells can grow more, or they may go to a next stage of the cell cycle. Okay, so those things you need to consider. Then what are the appropriate reagents, drug concentration for this assay? So some small scale optimization assays required to optimize your drug concentrations and all. Then regarding the objectives and images per well. So what is the resolution you need? So you need to define the objective, because generally this microscope is available with 10x, 40x objectives, and generally higher magnification objectives are not recommended for these applications, like the 60x and the 100x, because they provide very small field of view and they also need very small working distance. So generally the large working distance objectives are good for these applications. So how does this affect feasibility of the experiment? Then how frequent is my subpopulation, and how many images are sufficient for statistical power? Okay, so this is very important. Like you have to define that how many images your software will automatically take per well, and from where you can get the enough cell counts for statistical calculations. Then biological and statistical relevance, that do my experiment design decisions accurately address my question? Have I normalized my subpopulations or populations? Does my data hold up to statistical rigor? And plating and preparation. So all these parameters you need to, on a small scale experiment, you have to optimize first before going for the high throughput imaging experiment. So an efficient high content imaging system should have high speed camera. Okay, because you have to capture multiple frames. So faster the camera, uh, lower the time requirement for the imaging per plate. Okay, then low exposure time. So lower the exposure time means camera can image faster. So because so many images needs to be captured, so if the camera is slow or if the exposure time is long, then camera will take more time to take one image, so eventually your experiment will get delayed. Okay, then bright fluorescent signal. So obviously if the bright fluorescent signal is there, then you can work with low exposure, but if your fluorescent signal is very weak, then you have to go for the high exposure. Then low magnification, because lower the magnification, larger the field of view. Large camera FOV. The bigger the camera sensor, the larger the FOV. So these days the SCMOS camera provides large field of view. So that's why the SCMOS camera are preferred for this kind of applications, because they are faster as well as they provide the large field of view. High numerical aperture increases the microscope resolution. Less fluorescent channels, minimum of 2 fluorescent channels to recognize the essential cell phenotypes. Discriminatory positive and negative controls. Rapid autofocus. So autofocusing is very essential, because the overall microscope optics, your objective lens, your sample will keep moving. So when it moves, the plate moves from one sample to other sample, it may leave some kind of a z drift. Okay, so some z drift correction mechanism or the autofocus modules are required, so that every time before imaging, the software can automatically focus the cells first, and then it can proceed with whatever the single focal plane or the Z-stack imaging you have defined. So precisely moving stage is required. Then environmental controls are required if you want to go for live cell imaging, but if you want to go for the fixed cell imaging, then the environmental controls are not required. Then ease of analysis for large data sets. So slow analysis will bottleneck will be the bottleneck for the system. So fast image processing and data analysis is required. Okay, the goal is to maximize data output and experimental efficiency to detect and quantify critical features from hundreds or thousands of samples in a particular conditions as fast as possible. So some more information on the objective lens and the cameras for high content imaging, like what kind of objective lens are preferred and what kind of cameras are preferred. So low magnification objective lenses are better for HCI applications, as they provide large field of view and results in more data or cells per image. And using oil or water immersion objective lenses add challenges to automation, because every time when this objective lens moves from one sample to the other sample, you cannot go and put the oil there. So that's why I try to avoid oil immersion or the water immersion objective lenses. Instead, use the long working distance objectives, air objective lenses. And S-CMOS cameras are preferred because of fast acquisition speed and large field of view. Drawbacks of this technology. So performing HCI is time consuming and expensive, and it requires a large volume of digital storage, several terabytes per experiments. Okay, because it will collect so many images from so many different wells, so it will be a huge chunk of data. And it requires specialized software for microscope automation, robotics, image acquisition, and analysis. So that's it for high content imaging, and this is the last lecture of this course. I hope you enjoyed the course, and feel free to give us a feedback, and if you have any question, feel free to contact us. Thank you so much.